Hi, I'm Jude Cobbing from ProWash. We took some water samples yesterday and we're here today to see what happens when they're analysed in the lab. Hi Erin, I'm Jude Cobbing. We took some water samples yesterday and we've kept them cool, just like you instructed. But I was wondering, would you be able to show us around your lab a bit and explain some of the procedures?
Sure, I'd be happy to, Jude. And welcome to our water quality lab. I'm Erin Ling. I'm an Extension Associate and Program Coordinator for the Virginia Household Water Quality Program here in the Biological Systems Engineering Department at Virginia Tech. This is one of our water quality labs at the university, and I'd be happy to show you some of the equipment and the methods that we use to process drinking water samples in our lab. Thanks, Jude, for bringing the samples in the bottles we provided and keeping them cool in a cooler box. I'll explain more about why this is important as we go. This is actually the first area I wanted to show you. It's where we receive samples. When a lab takes custody of a new sample, we make sure that it's in good condition and the proper temperature and volume. My colleague Asa Spiller is receiving the samples into our system. Labeling systems can vary from lab to lab. It's important to use the sample bottles and labels provided so that samples don't get mixed up. This is the first stop when we analyze samples in our lab. We use a method called ColAlert, and when we're handling microbiological samples, it's really important that we wear gloves and that we're very careful to avoid cross-contaminating any of the samples. So with this method, we collect the water in the container provided. We want to work with about 100 milliliters of water. We're adding the media substrate, which is part of the ColAlert package. It's basically bacteria food and includes some extra nutrient indicators that will allow us to know if either total coliform or E. coli bacteria are present. So after we've added the substrate, we shake the bottle, we let it sit for a little bit to actually allow everything to dissolve, and then we're going to pour the contents of the bottle carefully into one of these trays, which is called a Quanta tray. We send this tray through the sealer, allowing it to heat seal the back of the tray and keeping the water in each of these little cells. Following that part of the step, we actually put it into an incubator and it stays there for 24 hours at 35 degrees Celsius. And then we read the samples after those 24 hours. These are some samples that we analyzed earlier this week using the ColAlert method that are drinking water samples from Virginia. So you can see the range of presence. The bacteria make themselves known by showing up as yellow if total coliform positive. This first tray is showing basically no bacteria present because there are no yellow cells, which is what we want to see in drinking water. You can see the cells that are yellow in these two samples are positive for total coliform bacteria. And if we hold these trays under a UV or at ultraviolet light, it will show the presence of E. coli in the cells that glow. This allows us to calculate a statistical estimation of the number of bacteria in the sample per 100 milliliters of water and report that back to the client. We use the ColAlert method in our lab, but other labs may use different methods based on cost, regulatory needs, or equipment available. Now we're in another area of the lab. This is our Metrome Chromatograph, which we use to analyze for anions that are dissolved in the drinking water samples that we process. In our case, nitrate and fluoride. And so you might notice that there are these small vials here with about 10 milliliters of water in them each. These are actually subsamples of the bottles that you would provide to us. And it's important to go ahead and fill the entire bottle that we provide so that we have some extra volume to work with in case we need to reanalyze any of the samples for quality assurance and quality control purposes. We're in the area of the lab where we conduct analyses for pH and electrical conductivity. It's important that you also measure pH in the field because pH is a measure that can change with exposure to air or during storage time. ASA is running some of the pH and electrical conductivity analyses that we do in our lab. Overall, it's important that you maintain the samples at a cool temperature so that there's no change in the chemistry or microbiology during transport of the samples. Now I want to show you what a water sample looks like up close. We have a bottle here of pond water that we've collected, and to the naked eye, this water looks clear, like there's not really an issue. A lab would use a standard method that's approved for analyzing for microbes in the water, but we can use a microscope to zoom in and see those microbes that are in the water. It's important that you use the bottles that are provided by the lab, and if those bottles are being used for microorganisms, those bottles are sterile. So it's very important that you're using the bottles provided and that you're using a sterile bottle and following recommended sampling procedures to collect the sample to avoid any cross-contamination of the sample. We hope you enjoyed the tour of our lab and learning a bit more about some of the processes that we run behind the scenes. A lot of the things that we talked about analyzing for today can also be analyzed using field test kits. There's a place for both, but in many cases, lab tests are more accurate. We really appreciate the opportunity to work with you, and we're happy to hear your questions at any time. We're approachable. Help us help you.
This video was produced under the USAID Bureau for Humanitarian Assistance funded ProWash Award. For more information and to download additional resources on water quality monitoring and other WASH-related topics, visit fsnnetwork.org/pro-wash.